In the last two videos, I talked about glycolysis and the oxidation of pyruvate, which are the first two steps in cellular respiration. And in this video, I'm going to talk about the TCA cycle, which is also known as Krebs cycle or the citric acid cycle. And it is commonly drawn as just a large section. I'm just going to use one of these circles here. So we have this large circle here because it kind of happens in a circular process. So you have your acetyl-CoA up here that comes in from the oxidation of pyruvate. So this is your acetyl-CoA molecule coming in. And the first thing that happens is the CoA group leaves. So this CoA group goes off, and this binds with what is called oxaloacetate. So I will write that right here. I'm going to try and make this video as simple as possible because as you'll see in a lot of other videos, the Krebs cycle can be pretty daunting. Oxaloacetate comes in here and it'll bind, and the just the 2 carbons plus the oxaloacetate makes what is called citric acid. So why it is called the citric acid cycle. And I'm just going to kind of summarize the important steps. So right around here, early on, you're going to have 2 CO2 groups leave. So you're going to gain 2 CO2 groups, and then here, just below that, we're going to have 3 NAD pluses become 3 NADH plus 3 H pluses. And then here, interesting, we have our first ATP molecule since glycolysis. So right here, you're going to have an ADP plus a phosphate group come to make a molecule of ATP. And then something that we haven't yet seen is called the FAD, which stands for flavin dinucleotide, or flavin adenine dinucleotide, sorry. So it is, we're just going to abbreviate it as FAD, and it comes in and becomes FADH2. And then that is kind of the summary of, for every single acetyl-CoA that comes in. Remember, we have 2 coming in for every glucose molecule. You're going to gain 2 CO2s, 2 NADHs, 3 NADHs, sorry, a single ATP molecule, and a single FADH2 molecule. So I just want to summarize this once again. You have at 3 different points during the cycle, you have more reduction of NADH that form NAD plus that form NADH, and for every 1 acetyl-CoA that enters, you have 3 NADH that are made, and we also get the reduction of FAD to make FADH2, and then down here, this is actually a GDP that makes GTP, but that can be changed to ATP, which I've just written it as ATP to keep it simple. GTP is just used for a lot of cell signaling. And then the last key feature is when we regenerate the oxaloacetate. So the actual full cycle here is an 8-step process. So there's a lot of different breaking down of different molecules that happen, and there's a lot of intermediate steps here. So you'd have something like isocitrate, and you would have succinate and fumarate that are all made at different points during the step in the release of these CO2s and the generation of these 3 NADHs, and all of these different molecules that we're making. They're all caused by intermediates being broken down, where these molecules can come off, and then we end up with our oxaloacetate again. I just wanted to kind of summarize what we've done so far. So from 1 glucose molecule, we're going to write that right here. So from 1 glucose, how many NADHs have we made? If we include every single step, this is important, we have made 10 NADHs so far. So we've made 2 from glycolysis, so you remember, 2 from pyruvate, and then 6 from the TCA cycle. And then FADH2, how many of those have we made since they were just introduced in this Krebs cycle? For every one acetyl-CoA that comes in, we're making one FADH2, so we are making 2 FADH2 total for each rotation of the TCA cycle. And then how many CO2s have we made? So CO2s, we have made 6 CO2s. So we've made 2 from the oxidation of pyruvate and 4 from the TCA cycle. And then the big one is how many ATP, and I'll put /GTP here, have we made so far from one glucose, and it is 4. So we've made 2 from glycolysis and 2 from the TCA cycle. In the next video, I'm going to talk about the electron transport chain and chemiosmosis, which are kind of the key for making as many ATPs as we possibly can.